Let's dive into the fascinating world of DNA, where tiny molecules called nucleotides form the blueprint of life. Among these building blocks, cytosine often takes center stage, especially when it comes to gene expression. But did you know that cytosine can be modified in several ways to influence how genes are turned on or off? One key modification is methylation, where a methyl group is added to cytosine, creating 5-methylcytosine. This process acts like a dimmer switch, often silencing genes when methyl groups are added to CpG sites, regions rich in cytosine and guanine. To study these methylation patterns, scientists use techniques like bisulfite sequencing, which can tell us exactly where methyl groups hang out in the DNA. But methylation isn't the only game in town. Cytosine can also undergo hydroxymethylation, forming 5-hydroxymethylcytosine, or 5-HMC for short. This modification is like a stepping stone in the process of DNA demethylation, which can reactivate genes. Enzymes called 1011 translocation proteins, or TETs, play a crucial role here by oxidating 5-methylcytosine to 5-HMC, often using cofactors like 2-oxoglutarate and dioxygenases. Interestingly, hydroxymethylation can be detected using specialized techniques like TET-assisted bisulfite sequencing, allowing researchers to map these modifications precisely. These epigenetic markers can influence gene expression by recruiting binding proteins or methyl CpG binding proteins, which interpret the methylation signals. But DNA isn't just about methylation and hydroxymethylation. It can also suffer from oxidative damage from lesions like 8-oxoguanine, also known as 8-hydroxoguanine, which results from oxidative stress and can lead to mutations if not repaired. Enzymes like diamine DNA glycosylase help fix these oxidative adducts, maintaining our genetic integrity. Speaking of repair, the balance between methylation, hydroxymethylation, and DNA repair mechanisms influences complex traits, including behaviors linked to the brain. For example, regions like the hippocampus, prefrontal cortex, and limbic systems are involved in cognitive functions, executive control, and even circadian rhythms, the body's internal clock. On a molecular level, factors like brain-derived neurotrophic factor or BDNF are essential for neuroplasticity and cognitive health. The interplay of these epigenetic modifications and repair process shapes how our genes function in the brain, affecting everything from mood to memory. So, in the ever intriguing dance of DNA modifications, enzymes, and proteins, each plays a vital role in fine tuning our genetic and cognitive landscapes, all contributing to the complexity and wonder of life itself.